For those of you who are less familiar with DNA sequencing technology, I've prepared a few slides to give you a little bit of insight in how this works. To be clear, myself, I'm not a biologist, I'm a technologist, but I've used some public resources to make it simple and to make sure that this kind of information can be shared and also can be accessed by you in case you're interested. DNA sequencing is the process of determining the nucleic acid sequence. I think that's all known to you. The nice thing about DNA is that it basically consists of four building stones, 4 bases to be exact: adenine, guanine, cytosine, and thymine. Those 4, as you can see in 4 different colors, can be sequenced and looked at on a molecular level. And that's the nice thing about sequencing technology. If you have those 4 bases that you can address specifically, you can determine the sequence of those bases relatively straightforwardly using different techniques. And in this way, you can basically sequence and decode an entire genome. And that's what sequencing is basically all about. And you can say that the advent of rapid DNA sequencing methods has greatly accelerated biological and medical research and discovery. And that's exactly where we come in with our microtechnologies, making consumable parts, for example, flow cells, to support this rapid uptake of DNA sequencing technology in the medical and diagnostics industry. Let's go back a bit in history. Again, I'm not a biologist, but I would like to at least summarize all the techniques that are there, and then I'll focus on the technologies that we use at Micronit. The first generation of sequencing technologies came in the 1977 timeframe, where Maxim and Gilbert started a sequencing technique called chemical sequencing. And also Sanger, I think that's the most well-known type of sequencing before next-gen sequencing came in, had developed a technique with chain termination. And with the Sanger sequencing, basically the whole human genome project was done, which was finished in the early 2000s, having costed billions of dollars and many man hours it took. And we all know that after that, things went very fast. Looking at what formerly is called second generation techniques, but better known as next generation techniques, we're looking at in general at technologies that look at short reads. So base pairs typically read in the tens to hundreds of a sequence, up to roughly 150, 200, or maybe 300 base pairs can be sequenced with this technology, and that's what we call short reads. The advantage of this technology, next generation sequencing, is that it generates a high throughput. And there's multiple technologies that have been developed over the 2 last decades, and I've tried to summarize them here below, including the companies that are using these technologies. Ion semiconductor sequencing used by Thermo Fisher, specifically in the ion torrent equipment. Pyro sequencing used by Roche, the 454 sequencing system. And one of the most well-known today next-gen sequencing technologies developed by Illumina, or I should actually say developed by Solexa, that was acquired by Illumina about 2 decades ago, is sequencing by synthesis, which uses the individual nucleotides that are fluorescent and can be imaged through an optical microscope one by one. Then there is CPAS-based sequencing. CPAS is an abbreviation for combinatorial probe anchor synthesis, which is used by BGI MGI, which is also a growing company based in China and now serving customers globally as well. Sequencing by ligation, used by the solid tools of Thermo Fisher, and electrical impedance-based sequencing, which is used by Genapsys, maybe a bit less known to you. As said, quite some technologies in the next-gen sequencing arena, and most of these technologies use some kind of flow cell or consumable device to sequence the DNA. Then there is what I would call the third generation. Mostly it's also added to the next generation pool of technologies, so that's a bit, let's say, arbitrary. But I've distinguished here the long read technologies, especially the one that has been developed by Pacific Biosciences, which is called single molecule real-time sequencing, where really individual molecules are sequenced in nanowells structures. And then there is nanopore sequencing, which is offered now by a number of companies, Oxford Nanopore, Quantapore, and Stratus Genomics. And they are basically using pores in silicon to sequence the nucleotides in the DNA, which is a different approach than using, let's say, optical-based technologies that use imaging. Today I will focus more on the next-gen sequencing technologies. Just to give you some examples of the current market leaders in next-gen sequencing, to be clear, these are just some platforms of market leaders that we see, not specifically coupled to us as a company. Illumina, I think by far right now is the market leader in offering next-gen sequencing approaches. They have a range of benchtop and high-end sequencing platforms that they sell to the market today. And they're using the sequencing by synthesis technology in all of their devices. Then there's Thermo Fisher, also mentioned already before. They have the ion torrent tools that were developed by, I believe, Life Technologies, which was acquired by Thermo Fisher. They have the solid equipment, so also quite huge in the market. And then there is BGI upcoming from China, as I mentioned before. And if we look at nanopore sequencing, which are now also addressed for now, the moment as next-gen sequencing, Oxford Nanopore is growing quite rapidly because they are developing a system that's really portable and is using, let's say, disruptive technology to sequence DNA. If we look at the market trends a bit, I have used some material from slides from market studies. In this case, I'm showing some material from DeciBio, a market report that they made in 2017, which ranged from let's say the time frame from 2014 to 2020 this year. And new reports, of course, are coming up soon. But the point I want to make here is that the market of next-gen sequencing is growing very rapidly. Right now, in 2020, this was anticipated 3 years ago, and I think the numbers are even higher today. We're in the multi-billion market range with annual growth of 15 to 20% on a yearly basis. What we also see, and that's why I added the graph to this slide, is that the markets are still very much based on either academia, but if you look at the growth, the clinical market is growing most rapidly. And that's also what we observe with our customers, that customers that are now these days engaging with NGS are really zooming into one specific diagnostic application. This is, I think, the most rapidly growing area within the next-gen sequencing field. Some other trends we see, I mentioned already, right now it's driven primarily by clinical applications, more specifically oncology, liquid biopsies, non-invasive prenatal testing, infectious diseases, think of the COVID-19, for example, which is very actual at the moment, and other rare diseases. But there's also new applied markets outside of the medical industry, which are the agricultural sector and biotech sector. Sequencing seeds, for example, for the crops is a really huge market to be engaged in. Forensics is a field, of course. If we all think about the CSI programs, they're using sequencing technologies to be able to act fast when there is a suspect that needs to be investigated. And then there's the biopharma industry as well. Still, about roughly 40% of the entire NGS market, as this report estimated, is driven by academia. And especially single-cell genomics and population genomics are two fields that are growing there. And the topic of single-cell genomics will be addressed by me in the second part of this webinar. Now, if we zoom into the products that we make as Micronit flow cells, this is another report that I took from Yole, April 2019, last year, which shows that right now the flow cell consumable market, specifically used for next-gen sequencing, is based on glass flow cells as well as silicon. And the interesting thing here, besides the absolute numbers, where we see strong growth from about slightly over 1 million of flow cells in 2018 sold to the market to over 4 million of flow cells anticipated to be sold in 2024. We also see that the balance between glass flow cells and silicon flow cells is slightly changing. Where glass is still one of the main materials used in building NGS flow cells today, we see that with the growing trends to develop towards CMOS-based technology, but also towards nanopore sequencing, silicon becomes more relevant as well. And you can see that Yole estimates that basically around 2024, those two materials will be evenly balanced. And that's, I think, an interesting note to make here. To give you some examples of the flow cells in the market today, I already brought up Illumina as being the market leader in next-gen sequencing. Here you see some of their flow cells offered. These are all taken from the report and from the web. You can find them there. But there's also some other examples of flow cells based on hybrid technology that are made for platforms that are out there in the market. Now let's zoom into some of the trends and then make the link to the technology. I think that's relevant to go through and to understand where this market is going, and especially for us to understand what kind of technology should we offer in the future to customers like you to be able to serve you with your product development. As I briefly brought up already, the market is growing and it's driven primarily by clinical applications, and this is an important note, because it means that we have to switch from a research use only environment to a clinical diagnostics environment. And this means that we have to start developing and manufacturing these parts in a different way, keeping in mind that at the end it should be CE marked or FDA approved to be able to use it for diagnostic applications. Also, another trend that is obvious, and you can see it nicely in the graph at the right side, is that the sequencing costs are rapidly decreasing. And usually the costs are expressed in terms of cost per genome. And the graph you see there is that starting from 2008, you can see a rapid decrease of the cost per genome, still going down. Right now we're in the $1,000 per genome range, but we know that the big companies out there like Illumina already have developed technologies that will bring down the cost towards $100 per genome. And this is needed to generate higher data output. And this means you have to optimize on your density inside a flow cell. If you look at flow cells, for example, where you're looking and imaging DNA clusters, you really want to increase the density to be able to increase a higher data output and to increase the number of reads you can make in one run. And one of the technologies that we can offer is using patterning technologies, either with microstructures, but more importantly, if you want to increase that density, you have to move towards nano patterning technology as well. Another trend we see in the market is that, and I've shown that already in the introductory slide, we're moving from next-gen sequencing into the field of long-read sequencing, where short read lengths no longer are sufficient to generate specific information, and it's really relevant to create long reads which provide new information to the customers, to the clients, and investigate new types of diseases, for example. And this long read sequencing is growing fast. Another trend we see technology-wise is, and I mentioned it briefly in the previous slide where I presented the glass and silicon flow cells, it's not only about just making a straightforward glass flow cell anymore. CMOS technology is hitting in fast. Where we have a base of microtechnology available to make a CMOS device, and usually the CMOS is then coupled to an optical glass lid and combined into a hybrid solution, which we call hybrid integration. The CMOS taking care of all the electronics and of the chemistry, the optical glass taking care of the microfluidics and the transparency that you need to be able to look inside the flow cell. And then last but not least, a trend already mentioned in the early start of this webinar, lab workflow automation. And for that, we also need to integrate hybrid solutions into one product. So moving forward to some of the trends, some of it briefly mentioned already. How do we approach this market, and also how do we see the future of this market in the next 5 to 10 years? As mentioned, NGS is moving from a research arena towards a diagnostics arena with all the implications it has. Then what we see is that whole genome sequencing has matured to an established and microfluidic based technology using optical flow cell consumables. It's not a highly experimental tool anymore, it's really common business. So it's really a common service to offer to customers today. Also, the rapid uptake of NGS for cancer diagnostics is a relevant trend that we see growing towards the future. And especially the area of oncology is the largest therapeutic area at the moment within NGS. But application areas like non-invasive prenatal testing are growing with the use of NGS technologies, being more accessible and also being more decentralized in labs rather than in central institutes that are doing high data output sequencing. Then more recent trends that I wanted to add, because also we're looking at the current situation, we see a rapid uptake of metagenomics, meaning that we basically take samples directly from the field that are being sequenced to, for example, monitor the mutation of viruses like COVID-19 across the globe. And I have an example of that in the next slide. And last but not least, if we look further into the future, it's not just about normal biology and medical applications anymore. It's also looking at synthetic biology applications using sequencing, especially if you look at data storage. And I gave an example between brackets here, which I took from the internet. In theory, all man-stored data up to today, which is about 40 zettabytes, can be stored into 90 grams of DNA if you use synthetic biology and sequencing techniques. And this means this creates a huge potential. If you look at the Internet of Things and all the data stored these days in the cloud, we need more storage. And DNA sequencing with the application of synthetic biology could be a very relevant area. And I see future growth there. I mentioned already the actual situation. NGS these days is heavily used for genomic epidemiology of the COVID-19 virus. And I took this picture from the internet. If you're interested, please, after the webinar, go to this source, nexttrain.org, and you can play the movie. And you can see how the virus, how it all started in December 2019 last year in Wuhan, China, which is the purple circle, and how it spread across the globe. And this can all be monitored very accurately with using next-gen sequencing techniques. And right now, unfortunately, as you can see, there's a lot of huge circles globally, and that's what we're struggling with on a daily basis. And of course, we all hope that this whole situation will decrease rapidly. I just wanted to show this to show the power of next-gen sequencing for studying these kind of pandemics. Very quickly going through some of the technologies for NGS that we use. And I have one slide of each technology before I close this part of the webinar. Pattern flow cells, mentioned before, hybrid integration, and lab workflow automation. For pattern flow cells, we make use of a combination of glass and silicon technology, where, for example, microstructuring of the bottom part can be combined with an optical lid at the top part, as you can see in the example flow cell presented here. Then with an optical microscope, if you make micro wells, in this case these were 1 micron wells, you can still see them with an optical microscope. Obviously, if you go down to the nanometer range, making wells of, for example, 100, 200 nanometers, you will need atomic force microscopy techniques or SEM techniques to be able to see the wells. But this is a technology that is growing quite rapidly to be able to store all the data into one flow cell. Then hybrid integration, one example of a wafer where basically we combine different materials together. The top part, which is transparent and can hardly be seen, obviously is glass. We can structure the glass with microfluidics. We can open up glass to facilitate the access of wire bonding, for example. And then in the center area of these devices, you see a central detection area, which could be a CMOS-based product, which could be a silicon-based product. But the message here is that combining a glass microfluidics part with a CMOS-based or silicon-based electronic part can be done by using hybrid technologies. And to come back to those technologies, the technologies we offer today, ranging from temperature, room temperature, all the way up to higher temperatures, or laser-assisted bonding, which is used commonly for functionalized surfaces, patentable adhesives, which is a common technique used for glass and combining glass and CMOS wafers, eutectic bonding techniques for bonding metals to silicon, anodic bonding techniques, which is used for bonding glass to silicon directly or glass to metal, and the most commonly used techniques when you have a single material to work with, for example, an all-glass flow cell, those can be directly fusion bonded using high temperatures. All the techniques you see in the slide are offered by us in-house. Lab workflow automation, as mentioned, this is a nice example of a device where you see a plastic cartridge that can be accessed. This is a microtiter plate format. It can be accessed fluidically through a robot pipetting into the side reservoirs. The microfluidic infrastructure will take over and will access a silicon sensor in the center area, which could be a flow cell, which could be a multi-electrode array device, or anything else. And in this way, you combine these materials and automate your workflow. So to conclude on the sequencing part, Micronit is a global player in the design, development and the manufacturing of NGS flow cell consumables. And we basically work based on customized designs, as you can see in the second part. We try to address the specific needs of our customers, building optical grade high quality glass flow cells, but also making glass silicon hybrid flow cells, for example. We have a strong base of innovative technologies, which I showed in one of the earlier slides, to make them microstructures and also nanostructures, also using various bonding technologies, electro and sensor integration, and patterning technologies.